Hey guys, so today we're going to talk about levels of structure in protein and in carbohydrates. So we're going to start off with carbohydrates, and I want you to remember that an example of a carbohydrate is a monosaccharide. And monosaccharides are single sugar units. So each one of these not bonded together is a monosaccharide. An example of a monosaccharide is a glucose molecule. When we start hooking together a bunch of glucose molecules, we can build what's called a polysaccharide. And we've talked about several examples of what polysaccharides are. There's a specific term to describe the bonds that occur that build a polysaccharide. And those are what we call glycosidic linkages or glycosidic links. Those are the bonds that hook together monosaccharides. We're going to focus specifically on glycogen, starch, and cellulose. So the first thing that I want to draw your attention to are the bonding that occurs between those monosaccharides. If you notice, you'll see a downward V shape in between them. That means that these molecules are very unstable, or in other words, that it's very easy to break those molecules apart. That's why we use these as energy resources to fuel our cells. Conversely, if you look at the cellulose bonding in between those monosaccharides, you'll see an alternating pattern. You'll see some downward V's and some upward facing V's. Because of that alternating pattern, that can make the molecule very stable, meaning that it's not very easy to break it apart. That's why some animals cannot digest cellulose, i.e. humans cannot break it down, that material that's found in the cell wall of plants. It's because of those alternating linkages or in that secondary structure of carbohydrates. I want to talk now about proteins and how we can have different levels of structures in proteins. But before we do that, we need to figure out how do we build a protein. Well, remember that proteins are made up of amino acids. And when we hook together a bunch of amino acids, we can get what's called a polypeptide. That polypeptide will then undergo a bunch of folding in the cell that will determine its function or its job. So first look at each of these amino acids. This is one, this is two. If we want to hook together those amino acids and form a peptide bond, a process has to occur. And that is dehydration synthesis, meaning taking away a water molecule to put together two amino acids. There's some directionality involved, meaning that it has to occur in very specific spots of the amino acids. So if you look here, you should recognize those as functional groups. This is a carboxyl group, and this is an amino group. If the carboxyl and amino group are side by side, dehydration synthesis can occur, meaning taking away a water, and a peptide can form, or peptide bond can form, holding together those amino acids. And that's how we build a polypeptide, and then eventually a protein. So what I just described to you was the primary protein structure. That is the linear flow of amino acids hooked together by peptide bonds through dehydration synthesis. Then the protein will undergo a secondary folding, and it will determine its secondary structure. It can have two options, either an alpha helix shape, which is kind of coiled, or a beta pleated sheet, which is almost like alternating bends. Some proteins can have some of both. That secondary protein structure is the result of hydrogen bonds. Some more folding that is going to occur, and this is going to be the tertiary structure of the protein. And this is probably one of the most important folding activities that occur, because the tertiary structure determines the overall three-dimensional shape of the protein. So far, we've been talking about something that is almost very linear, and now we're going to start talking about the actual folding that occurs to make it three-dimensional. And these tertiary structures are the result of a bunch of interactions. Things like hydrophobic interactions, van der Waal interactions, R groups interacting, In addition, the tertiary structure is responsible for the specificity of the protein. So specificity refers to the protein's very specific shape and the job or the function it's going to perform. Lastly, we can have a quaternary protein structure, and this is when different polypeptide chains start bonding together. So if, let's say, for example, we're talking about hemoglobin, that final quaternary structure of the hemoglobin is the overall polypeptide chains, 4 of them, bonded together to give it a very three-dimensional shape. And then you could dissect that protein to its tertiary, secondary, and primary structures. So why are we concerned with protein folding? Well, first of all, there are proteins that assist other proteins in folding, and these are called chaperone proteins. Those proteins that help and assist proteins in the folding process. If those proteins don't work or fold properly, or those chaperone proteins are not present, you can get what are called prions. And prions are proteins that have not folded correctly. So prions. And those prions are thought to be responsible for things like mad cow disease and Alzheimer's. And so there's a lot of research currently going into targeting those prions and either fixing them or eliminating them without causing harm to the organism that they're studying. In addition to the folding, there are many ways that we can unfold a protein or denature a protein. And the ways that I want you to be familiar with, the first is temperature. Changing the temperature can interfere with some of the hydrogen bonds within the protein. Changing the pH can also affect some functional groups, and it can affect some more bonding that can occur. Or you can add more concentration of something, specifically polar and non-polar substances. If you add polar substances into or around a protein, it can interfere very specifically with the hydrogen bonds that are holding together that protein. If you increase the concentration of non-polar substances, they could interfere with those hydrophobic interactions if those are the types of interactions that are holding the proteins together. So it really just depends on which protein you're talking about and what type of interactions are present. And that will determine what factors can denature the protein.